Hey everybody, welcome back to HPC Tech Shorts, where every week we take you to the water cooler here in HPC Engineering at AWS, and we talk to the engineers, the architects, and the scientists who are helping make the cloud the best place for your HPC workloads. In a couple of the past episodes, we've talked a bit about Nextflow and other sort of HPC pipeline tooling software stacks. And in particular, we got into a good discussion with Evan Floden from Sakera Labs, one of the core coders on the Nextflow project. And we talked a lot about how Nextflow's helped help different parts of the community, particularly during COVID, where cloud computing and the ability to spin up these workflows at scale very quickly. In response to a global crisis, turned out to be, really, really important. This week, one of the things we discussed with Evan was NF Core, and so I'm actually really excited that this week I'm joined by Phil Ewels from the SciLife lab at Stockholm University. Hey, Phil.
Hey, nice to be here.
And my colleague Angel in Philly from the developer relations team. Hey, Angel.
Hey.
Now, both of you guys know about 1,000 times more about genomics than I do. So this is going to be more of a discussion for me to be the... I'm just going to be the good-looking guy in the sidecar watching. Maybe I'm going to fail at that badly. Anyway, Phil, you're one of the founders of the NF Core project.
Yeah.
Fill us in. Where does NF Core fit in that picture?
Yeah, so I mean, NF Core built on the foundation that was laid by the guys building Nextflow. Like most of the projects I've been involved in, it kind of came around by accident. I just was building something we needed. I work at the National Genomics Infrastructure in Sweden, which is part of SciLife Lab, and we run analyses for lots of different types of data. And we wanted to go with Nextflow because it was clearly very good at running at scale, and figured it would future-proof us for moving to the cloud or to other systems. And so we started building pipelines. We started kind of standardizing them, and because Nextflow is so good at being portable and reproducible, it kind of took off, and it took me by surprise. And we started reusing the code that we're doing in-house. It started spreading to the people we were delivering data to in Sweden, but then other facilities around the world started picking up our workflows and running them as well, which is testament to just how portable these pipelines are.
And where were they before? So this is before you had NF Core as a project. Where were they picking the workflows up from? Was it just emailing you, or?
No, so I mean, for our workflow at NGI, we do everything in the open on GitHub. So all of the code was there, and I'd been going to the Nextflow meetings since they started. So I'd been presenting some stuff there. So there was a little bit of visibility as the nascent Nextflow community started to grow. Mm-hmm. And then I think it was a second or third Nextflow meeting where I kind of started to really actually put down on paper my ideas of how it should be done. We started building a template and started writing some guidelines for ourselves, and got chatting in a coffee break to a couple of people, Alex Peltzer in Germany, and we started collaborating with them. And he was like, "Hey, we want to build this new pipeline. Can I call it like SciLife Lab NGI something, just so it like fits in with the rest of your pipelines?" And I was like, "This is crazy." So maybe we should take a step back and strip off all the SciLife Lab stuff and say, "Okay, let's properly collaborate on these and call it something new."
I remember getting my. I think I was there at that meeting in Barcelona when you guys were first announcing NF Core, and I got my NF Core sticker, and I think that was it, wasn't it? That was the meeting where we had was that the one where we had the innovative collaborative cooking?
Yeah, that's right. The tapas.
Yeah, the collaborative tapas, where we all took turns to create dishes for each other, and there was also a group creating innovative cocktails, and there's a whole lot of stories that come out of that. I remember far too many of those.
Yeah, those before times were really tough on you, right?
Yeah, that's right. But this is what I find fascinating about, like, if anybody out there has never been to one of the hack fests that the group in Barcelona puts on around Nextflow, go to these things, because you learn so much, you see so much, but it's kind of like I don't know, out of all of the open source groups that I get to work with, and that's a lot, this is the exemplar of how to form a community and get some really awesome things coming. And I mean, NF Core is kind of it's the cherry on the cake, I think.
Yeah, like I say, I mean, it was a happy accident, and NF Core doesn't, I mean, we do some cool stuff, but the thing I cherish most in NF Core is that community, is the networking and the people. That's what makes it interesting, and that's what makes it fun to work with. And all of that it came out of, like you say, that meeting kind of was very good at bringing together like-minded people working on the same set of problems. Yeah. And then the talks are great, you learn a lot, but exactly throwing everyone into like random situations and getting everyone chatting is really good.
And we will do it again, and I promise I'm going to be there to buy you a drink. But there's a serious side to this too, because like when the pandemic happened, suddenly everybody in the world wanted to know about spike proteins and all of the things associated with that. And there was quite a bit of interest in, obviously people needed to spin up infrastructure real fast, and they needed to get answers very quickly. And it was like, blow out all of the barriers. What do we need to get this done? And there's a story there, right?
Yeah, exactly. I mean, pretty much everyone I knew working in bioinformatics went over the period. We actually had our last NF Core meeting. We have NF Core hackathons. We had our last in-person one in London at the Crick, right on a knife edge as to whether we were going to hold it or not, because this whole new COVID thing was kicking off. And we got back, and it was clear that this was going to be a major thing. And almost everyone I know just had to drop all of their projects and be like, right, you don't know anything about virology, but you need to build something that will work here. So very, very quickly, we mobilized a group of people within NF Core who wanted to kind of work together on this, because we figured that we were really well placed. And you saw this happening across the community. Like there are Nextflow pipelines popping up all over the place. There's a very well-used Nextflow pipeline. It's not NF Core, but it's building on Nextflow, which was used in the UK a lot when they were kind of springing up. And we got together. There's a few groups, so London and Spain were some of the major groups contributing to this, and we put together our own pipeline for doing this kind of analysis, which is called viral Recon.
So hang on, let me. I've got to click on a button here. You've got viral Recon to show us. Tell us more.
Yeah. So this pipeline, like I say, like much of NF Core is very much a community effort, and I haven't had that much involvement in writing this pipeline, but it's really exciting, and I've used it a few times now. And the idea basically is that you take sequencing data for the COVID genome, so either Illumina short read or Nanopore long read sequencing data. And it supports a whole bunch of different preparation kits. So whatever's been done in the lab, all these different ways of doing a similar thing, it should support most of them. And then you throw that data in, and it takes you from the raw data, which is just a bunch of ACTGs, through to something which is actionable. Which in this case, the main thing is looking at the variance against the reference. So then you can say, okay, is it this stream or this strain or that strain? Is it the Delta variant, or is it is it this or that? And that came together so fast. It was quite amazing. I mean, we were motivated and everyone on the same page at the same time, and this pipeline just like flew together. It was really nice to see. And it's being picked up all over the place now. So it's being used routinely in quite a few different countries, and every now and then see kind of tweets and stuff, like one the other day about it being used in Colombia to look for different variants and stuff. So I hope it's had some impact. And like most of the NF Core pipelines, got some really nice QC metrics, which makes up a large part. So we've got this nice heat map here, which kind of shows how well the lab part of the protocol is working across different samples. And
you totally need to turn this into English for me. What is that? It looks cool. What is it?
It looks cool. It's like the Matrix. No. So, well, this plot in particular, you've got the rows are samples. So each row here is a different sample, a different input sample, so a different person. And then in the columns, so if you imagine the COVID genome as a long list of letters that make up the DNA, you call that a reference, and you can say, okay, it starts here at 0 and it goes through to, I don't know how many 100,000. When you analyze that in the lab, you start from very, very tiny amounts of DNA or RNA, and you need to amplify that so that you've got enough of it to actually put onto a sequencing machine. And typically that's done by using a technique called PCR, where you pick specific parts and you kind of amplify those parts. So each one of these columns is a different primer pair. It's a different part of the genome that's amplified. And if it's brightly colored, it tells you it's present and it's worked. And if it's not, then that one hasn't been picked up so much. So you can see this particular primer pair is kind of working less well across all samples. And you can see that these samples just haven't really worked. They probably don't have any COVID sequence in them.
Mm-hmm. Yeah, and this is
everybody in the world
important.
knows what a PCR test is now. So,
getting QC information quickly is really important for high scale and high throughput lab operations, because sometimes you can recover that sample. Sometimes it just means that the sample is bad, you have to throw it away. But other times you can put the sample back onto the machine to get more coverage. Obviously adjusting for any batch effects that you might have from library prep. But this at least will tell you, in the grand scheme of things of garbage in, garbage out, you don't want to put garbage into your analysis pipeline. So upstream QC metrics are really a pretty important thing to make sure that you're recovering from batch effects, or B, is this total garbage? Then you really shouldn't move forward with analyzing it.
Absolutely.
Right. Got it. Okay, this is cool. I tweeted this the other week that the stuff that you guys are doing in the life sciences space, in particular the impact that you've had on COVID, that's the stuff that gets me out of bed every morning and makes me want to come to work and feel good about what I do. Is doing stuff that helps groups like this do this. So.
The other thing which I really like about the workflows that we build, I mean, Nextflow itself is a command line tool. Generally, you run Nextflow on the command line and it kicks everything off and it does all the clever stuff. And there's a bit of a learning curve to using Nextflow in this way. You have to kind of know the ins and outs and rewrite we put a large focus on documentation and stuff like this. But one of the things we've done within NF Core as a community is started to standardize how we describe some of the ways that the pipelines that we build work. And we're trying to feed this back into the main Nextflow community now. And the first thing we started with was a way to describe all of the parameters, so the different flags that you specify on the command line, like describe them in a computer readable way, machine passable. And that allows us to build like really nice interfaces like this big launch button. Which I love. So if I hit this, then you can get taken to a nice web GUI. And now all of these command line flags that you'd have to run instead kind of nice little text inputs where you can type it into your web browser. You've got help text like embedded alongside it. So you don't have to have that whole kind of learning curve of figuring out and going and reading pages of documentation. You can kind of just throw yourself in and it checks. So like, well, it should do. It checks to see whether you've, if I put in like a dodgy email address or something, it should tell me that something's gone wrong, and let's see if it works.
Right. So at the end of this, when you say launch, does it launch it, or does it produce like a command line that you copy and paste? What's the end result here?
So within NF Core, we try not to kind of tie ourselves to anything. We try and keep it as flexible as possible. So you've got three different options here. Mm-hmm. You've got the Nextflow command here, which is actually super boring because I didn't put anything in, but so it's just running with the defaults. So you can just copy and paste whatever it gives you and put that into the terminal yourself. No edit software or anything. We've got some helper tools which we've written ourselves. So we've got our own command line tool which wraps around Nextflow and does cool extra stuff. So in that case, you can copy this and it basically does the same thing. And also, I think Evan mentioned Nextflow Tower, we've got a nice button here. You click that and it takes you off the next flow tower with all of those things filled in.
Wow.
And in fact, the launch interface that Evan showed in a previous talk is built off the same underlying schema. So we kind of work together on that, and that's the beautiful. Rather than kind of hard coding something into our website for this, we developed a standard, a schema, and then the tools go on top of that, and then anyone can build on that.
The layering and the integration here is awesome. Now, Evan's actually going to come back very soon and do a talk about Nextflow Tower, just exclusively on Tower, and just give us all the demo. And just the little glimpse that we got last time was awesome. It looks really cool.
I want to shift a little bit of conversation here, if that's okay with you both, because I'm seeing some really excellent things that I want to dive a little bit deeper into. And one of them, Phil, you brought up about building community. You know, like technology is one thing, but then bringing the community along and building that community to form shared best practices is a lot of work. And I'm seeing a lot of things in NF Core that you know, go towards that goal, specifically, you know, your usage information, your developer documentation. Can you go through a little bit of what, you know, the types of things that would be needed from a user perspective first, and maybe from a developer of pipelines perspective first, that would help you be part of this community, a part of this NF Core community? So let's start with users first. Where should they go first? And I mean, we just saw an example with the viral sequencing, but what about in that top layer of pipelines and content and like finding the right pipeline? How would they go about that?
Sure. I mean, our homepage is kind of, that's what we try and centralize all the information there. Underlying this, everything is on GitHub with open source licenses, but it's spread across, I don't know, 70 different repositories. So you have to sort of know where to look to find that. But the website brings all of that together, scrapes all that content and sticks it all in one central location to try and make it more findable. As you said, I'm kind of a bit of a hardcore fan of writing documentation. I try and put a lot of effort into it because I think it's. And
were those metadata tags? Sorry, I'm gonna put this back up here.
Weird developer right there. There's
like if you go to one of those pipelines, like if just pick a pipeline or view pipelines. I want to see a couple of these things. You have things like metagenomics, 16x. Where do those metadata tags come from that sort of power that search engine and keywords?
Very simple. It's if I go to the GitHub repo for this pipeline, there you go.
Okay, so they're right there. Okay. Awesome.
Wherever possible, we just hijack underlying systems. And GitHub, I love GitHub. So we just use everything that GitHub has on offer.
He loves version control and he loves documentation. We need to clone this guy.
Not just version control. I mean, GitHub Actions and automation and metadata.
Oh, you're getting me all heated up and excited here.
All right, so we got that's pretty awesome. So users could go through, they could find if they're doing RNA work or doing or looking for specific tools or domains. They can go through here and find it. Obviously, everybody looks for GATK probably as a first thing.
Yeah, exactly. Does anything come up if I search for GATK? Yeah, there you go. There's our main GATK pipeline.
Right. There's a couple of GATK based pipelines. And then as a developer, so now you know enough about this and you need to make, you want to share with the community. It either goes with a publication or you want, some specific collaborator program and you're a developer. Where do you go then? Let's look at some of these best practices that you guys have built.
Yes. So the first thing I'll mention is something we have on the homepage, which is that I think I try and advocate and try and get this out there as much as possible, which is that we're a bit different to other websites where you might kind of list a pipeline that you've built or list a tool that you've built, because we're a community from the word go, which means that you develop with the NF Core community rather than bringing along a pipeline that you've built. And that's really important because there's certain stuff which is a bit non-obvious. We have fairly strict rules about things like using our template and doing things the right way. And it's important that you get that right from the start. So the first thing I'd say is if you're at all interested, come and join the community first before you start writing any code. And then, we have guidelines. So if I go under developers and then guidelines, we've got kind of the rules, the rule book, where we sort of say, okay, what's okay, what's not okay for workflows which are part of NF Core. It has to be open source, has to use Docker and GitHub and stuff. And then after that, you hit the big button. If you're still interested at that point, then you hit the big join button and come and chat to us, basically. And so we use GitHub for all of the code and all the tools, and we use Slack for all of the organization. We're very funded, got some grant funding from Chan Zuckerberg, who pay for our Slack subscription, and so we have, I don't know, thousands of users now, I think.
Wow.
Awesome. How many people? Let's find out. And of course, numbers, I've got it in the tab already. Slack, here we go. We've got, going on 1,800 users now on Slack.
That's amazing.
So, I mean, it's a fantastic place, both if you're just trying to use a workflow or if you're a developer as well. And you go in, and we've got enough people now that there's basically always someone online, whatever time zone you're in, or whenever you're working, if you ask a question, you'll probably get an answer fairly quickly. It's a fun place to hang out.
That's fantastic. If people want to go, they can go to NF Core, the website. It's there and available for you to find all of this information. That's great. Do we want to do a little demo or?
Yeah. And
what do you guys think?
I do, and I'd like, I kind of want Phil to just explain what that little logo on the results tab is on the pipeline pages as well.
I skipped over that bit. So the page I was showing a second ago, but we have a dedicated web page for every pipeline that's available. So we have over 50 different pipelines now. So we came, our background, I work at a genomics infrastructure. Our background, most of us came from genomics. So we definitely have most of the pipelines are around genomics, but we have pipelines coming in now from other corners, more and more, proteomics and imaging and stuff. And we're kind of keen to get other people on board as well. But every pipeline we have has a web page, and certainly the ones that have got a release already, at least we've got a stable release, will have a nice little tab called AWS Results. And this is where you guys come in and help us out. What we do is when a pipeline is released, we automatically kick off a test benchmark run, which uses like a full-size, realistic published data set. And it runs on AWS Batch and saves all the data in S3. And then because we base the folder structure on the predictable Git hash, we can then tie that into the website. So what we're looking at now, we can just browse through basically a live, this is just looking through an S3 bucket, which is public. And we can kind of look through these files and see what the pipeline generates. So that report I showed you before is the multi-QC report. I click on that and it just kind of loads up in the web browser for me there. And all of the results, it's not just the reports, all of the results are there. So that means between different versions, you can benchmark what's changed. And if you're interested in running a pipeline, you can check out what it produces without having to go through all the faff of trying to get it to run yourself. You can just kind of go and poke around straight away and see what's in the results folder.
And that happens for every version of every pipeline.
Yeah. And the other thing is, in the early days of NF Core, most of us were running on HPC, and there are some things which are a bit specific about running on cloud because of its very distributed computing. So the other thing is, because we run every pipeline on the cloud now, we're certain that they work on the cloud, which is kind of nice. It finds the edge cases for us before someone else does.
Awesome.
Yeah, well, that's going to help our customers too, because then they don't need to worry about that faff. But it's helping everybody, right?
Those continuous integration tests are so vital for reproducibility.
So he likes.
Just development and forming the community, as well as getting new members in to say, okay, we can trust these pipelines because they've gone through a rigorous process, and also take a look at the results. That's really pretty fantastic.
It is. So he likes documentation, version control, and CI/CD pipelines.
Are you sure? Are you sure you're a scientist and not a developer?
Yeah. Right. Do we have a Colin, you guys are the genomics people. Do we have a cloning machine somewhere?
Not yet.
Nice. All right, let's demo. I think this is
Right.
we've got to do demo.
Yeah, what do you want me to demo? I'll quickly
Stick on
demo. Well, I was showing the launch button before. I was thinking I didn't show you the fact that you can also do this without using the web. So if you're running like on some hyper secure cluster that's offline, you can still kind of do that. So this is the NF Core, just a Python package that we have. So you can do like NF Core launch, and then I'll say the RNA-seq pipeline. But this could, again, we try and make these tools generic to any Nextflow workflow. So it should work with anything. You can just put in a URL to a GitHub repository that has a Nextflow pipeline in it, and it should work. And then just to show that we get this nice kind of, you can choose web-based, which is what I showed you before, or you can also do it on the command line here. So you can kind of go through, and again, we've got all the different kind of documentation built in there and stuff.
That's nice.
That's super cool.
A CLI, nice CLI workflow walkthrough, which is really
exactly, yeah. I love a wizard. So yeah, that's pretty cool. And the other thing which is kind of nice is that we have a lot of workflow tools here for developers. If I do NF Core help, you can kind of see all the different subcommands, and some of these are for, like the one I showed you, for kind of end users of a pipeline, but some are for people creating pipelines. So I can do NF Core create, test, and it will make an empty but valid pipeline for me. If I go in here, then you can see we've got all of the files created there. You've got the GitHub action workflows, you've got the README, you've got everything set up ready to go. And that's kind of the base of what we start. So that's an amazing way to get started with Nextflow, because you don't have to think about best practices. You've got a skeleton that you can just start filling in straight away. Mm-hmm. And then things like the parameterization, like you don't want to be messing around in this big kind of horrible JSON file, which is easy to get wrong. So what we do is we say schema build, and we've built some nice kind of web-based graphical tools for editing your JSON schema.
Phil, anything else we want to cover before we run out of time?
Yeah, one of the things that I'm quite excited about with NF Core going forward is the idea of the new syntax that's coming out with Nextflow called DSL2. And this allows us to do some really cool stuff to do with sharing components of workflows. So now the start of many genomics pipelines start with the same kind of tools that run basic QC on the raw data. And instead of everyone doing their own version of that same part of the pipeline, we can kind of have a shared repository of pipeline parts. And you can just import that in. And then if someone fixes a bug in that, then it kind of gets shared out. It's like if you're using modules in Python or anything else. And this is growing super fast now. We have like 160 different tool wrappers or something already, just within about 6 months of work. So if you're coming in building your own pipeline, maybe you have an in-house pipeline that you're never going to share with anyone else because it doesn't make sense. But you can still now come in, use the NF Core template to get started, use the NF Core modules install command, and pick all the different modules you want to use. And straight away, you've got like 6 different tool wrappers complete with the Docker containers that they use and all the code written up. And all you need to do is then just stitch them together. So you can build a pipeline like so fast. And soon we'll have like sub workflows as well. So you'll be able to say, okay, genomics QC, and it'll go. So this reusability of code between NF Core pipelines, but also for any pipeline developer, is super, super powerful. And we're just kind of about to hit go on that. So
wow, that is really okay. You guys just you keep doing stuff that's pushing the edge here. This is really cool.
No, this has been great. Always happy to talk to the Nextflow community. Like I've said before, you guys seem to be on the perfect cusp of there's a technology that's out there that's applicable to bioinformatics, and you're an early adopter of it, and also bring along the community to help you, not just implement some cool technology for technology's sake, but are good editors of saying, actually, this is useful, and show the way forward for the rest of the community. So a lot of props. Big
props. But everything from the bottom up, from the containers through to Nextflow itself, through to NF Core, every single one of those layers is removing undifferentiated heavy lifting. Okay, that's why we're so keen about it, because it aligns with what we're trying to do too. We want a scientist to be able to spin up scientific workloads without all the fuss and without having to know about how to buy a cluster to do it. So kudos, well done. Seriously, what you guys are doing is important, and it really makes a difference.
Thank you very much. And like I say, it's very much a team effort with hundreds or thousands of people now. So I'm excited to talk about it, but it's certainly, I can't take credit for much of it.
Well, people should go here for sure if they want to be part of this community, whether that means just using one of the pipelines and deploying it, or getting in and putting some muscle into building the community and doing things even better. And then the last thing I should say is, if anybody at home is watching and there's ideas that you want to see us cover and deep dive in future Tech Shorts, DM us on Twitter. Our DMs are open. Always happy to hear new ideas and suggestions. It's where most of these talks are coming from. But for today, Phil, Angel, thank you. Easy.